My name is AJ Holmes, and my project is the generation of CACNG2A mutant zebrafish using CRISPR-Cas9. My project started out looking for genes associated with eye tracking dysfunction, or ETD, a very consistent set of oculomotor deficits seen in patients with neurodevelopmental disorders like schizophrenia and autism, as well as their first-degree relatives. And the high penetrance of ETD suggests that genetic factors play a large role in its development. Several genome-wide association studies have identified potential risk genes for the development of ETD, and among those is CACNG2, which was identified as the most significant correlate of anicotinic error, an inability to inhibit reflexive eye movements towards stimuli entering a visual field. Knockdown studies have suggested that CACNG2A, the zebrafish ortholog of this gene, is functionally identical to the human gene, making zebrafish an ideal model for examining the effects of this gene on eye tracking as well as neural circuit formation in the visual system. To examine its function, we decided to create a line of CACNG2A mutant zebrafish using CRISPR-Cas9. In Figure 2, we can see the general mechanism by which Cas9 induces mutations. First, it's guided to its genomic target by the short guide RNA, or guide RNA for short. It then induces a double-strand break in the target sequence, and the cellular repair mechanisms will ligate the strands back together, and they might introduce insertions or deletions into the sequence, which will hopefully result in missense or nonsense mutations and loss of gene function. Then over in Figure 1, we can see the target sites that I've tested. Specifically, there's this exon 1 target site, as well as these two promoter flanking target sites that were designed to excise the promoter from the genome and result in complete loss of transcription of the gene. So we cross pairs of transgenic zebrafish that have GFP expression in the neural circuits projecting from the eyes to the brain, so we can study the morphology of these projections in our mutants. The embryos were then injected at the single cell stage with Cas9, the guide RNA, and GFP, and the next day embryos were screened for ubiquitous GFP expression to verify integrity of the injected RNA. A sample of GFP-positive embryos as well as uninjected controls were lysed to extract genomic DNA and fluorescent PCR amplified DNA fragments containing the guide RNA target site and tagged them with an M13 fluorescent primer. The fluorescent PCR product was then run using capillary gel electrophoresis, and fragment analysis was used to measure Cas9 activity. The injected Cas9 continues to cut at the target site as cells divide, leading to mosaic mutations throughout the embryo, which makes these injected fish poor models for examining CACNG2A function, so they are currently being raised, and their offspring, which will have individual mutations, will be screened using topo-TA subcloning and Sanger sequencing to identify loss-of-function mutants. Because these injected embryos are mosaic, capillary gel electrophoresis, which separates DNA fragments by size with high fidelity, was used to identify a variety of insertions or deletions produced by Cas9 as a cluster of differently sized DNA fragments near the size of the wild-type amplicon. In both Figures 4 and 5, the wild-type amplicon is denoted by these red asterisks, and when we're looking for Cas9 activity, we're looking for these cluster peaks surrounding the wild-type amplicon, something that's clearly missing in these uninjected controls from Figure 5. So prior to COVID, I had focused exclusively on the exon 1 guide RNA, but wasn't able to gather any fragment analysis data before suspending my project. And when I could pick things back up in the fall, my focus shifted towards the promoter flanking guide RNAs, which after multiple runs of fragment analysis both failed to show any apparent Cas9 activity. While this was unfortunate, fragment analysis data from a single surviving fish injected with the exon 1 guide RNA showed promising but inconclusive results. And in a bout of last-minute desperation, I did a large run of exon 1 guide RNA injections to try and see if I could finally get some samples with clear Cas9 activity. Unfortunately, of the 15 injected samples prepared for fragment analysis, 12 of them showed clear Cas9 activity, with these cluster peaks surrounding the sides of the wild-type amplicon that you can see in Figure 5, which was awesome. After generating our CACNG2A mutants, we're currently planning on measuring the optokinetic response, a test of reflexive eye tracking, where this gradient of lines rotates around the fish, and video of the fish's eye movements tracking the gradient can provide data on smooth pursuit and saccadic eye movements. We also plan on confocal imaging of retinal ganglion cell axons connecting the eyes to the brain to examine potential changes in the visual neural circuitry caused by this mutation. Our lab is also very interested in the homeobox transcription factors GSX1 and 2, and coincidentally there's several regions at which GSX1 and 2 could regulate the expression of CACNG2A, which may have implications for deficits in prey capture that could be due to abnormal eye tracking in our GSX1 mutants.